Okay, good. Now you can hear me. The couple of questions, or the last question here, actually gave a nice bridge to my talk. Can I see my talk here too? Talking about large scale and smaller oil spills. Wait a second. Yeah. A bit behind schedule, but I'll try to keep it shorter than the performance we had yesterday at the dinner. Very good. So this is what the general public usually thinks of when they think about oil spills. It's a large scale disaster, such as the Prestige disaster in 2002. But as somebody pointed out, not all spills are large scale, and actually far from. And what most people don't know is that there's a huge amount of small scale, illegal and quite intentional oil discharges going on every day. And the shocking thing is that the amount of oil that's spilled through these discharges greatly exceeds the amount of oil that we have in these kind of accidents. For instance, it's been estimated that each year in the Mediterranean, these illegal oil spills, illegal oil discharges, add up to nearly 20 times the Prestige accidents. So that's a huge amount, and they're done to save costs. Basically, it's tank washing residues and ballast water, all of that. So with this intro, it's very important that the authorities investigate every oil spill that they detect, and that's what forensic oil spill identification has to do with. So you want to link an oil spill to a suspected source and try to show that that's the source. And this includes a statistical evaluation. That's not all of it. That's a small part, but it's still an important part of it, is a statistical evaluation of how similar a spill sample is to a sample from a suspected source. And then the forensic scientists are called to court as expert witnesses to tell about their findings. And what I'm talking about today is some collaboration that I quite recently had with the National Bureau of Investigation Forensic Lab. So when an oil spill is detected, the Coast Guard goes out there and takes samples from the spill and try to identify ships that may be the source to this spill. And then they analyze those samples through gas chromatography. And here the left and right column show two different oils. And you see that the chromatograms, the dominating features of these signals are peaks. And each peak will correspond to a certain chemical compound. And the peak height is the abundance of that compound. And some 20 years ago, the comparison was largely done by visual inspection. And an experienced forensic chemist will actually be very accurate in saying whether we have the same oil or not. But since then, it has been realized that some sort of statistical evaluation would probably be helpful to even out the differences between chemists, and it's also very time-consuming to try to look at these chromatograms and try to sort of figure out. So they wanted something to support their findings. So the data that we get from chromatography is basically peak height measurements, and you'll have maybe 2 to 4 replications of these analyses. They're very time-consuming and costly. So this is very data poor. And the model is basically it's quite simple. You assume that the analytical variation is normally distributed, but there's a linear dependence between the standard deviation and the mean, which is lab specific, and that dependence maybe complicates things a little bit, but it's basically quite simple. So there's a statistical approach which is currently used, that's based on a standard issued by the European Standardization Organization, and what that says about the statistical part is that we should use frequentist hypothesis test with the null hypothesis being that the peak heights are equal, and the alternative that they're not equal. Well, so far good. So far so good. But then the reasoning is that if we fail to reject the null hypothesis, then that sort of supports. That's evidence in favor of a match. But that can't quite be correct, because hypothesis tests are actually only used to evaluate evidence against a null hypothesis, but not really in support of it. They don't make any reference to the alternative hypothesis. And it's I don't know, it's slightly perverse to go to court and have a null hypothesis which implies guilt, and say that, well, you know, we believe you're guilty unless we can falsify this hypothesis. I don't know, it doesn't sound very good to me. And the other problem was that it doesn't really result in a quantitative assessment of how close the match is. You can say, well, it's non-match or inconclusive, or maybe it's a probable match, meaning that we're not quite sure, but it's more than inconclusive. So the alternative approach, of course, is to do the Bayesian way, because then you can we can actually get a posterior probability for the null hypothesis, or any hypothesis, and you'd actually get a number, a quantitative assessment. And a quantity which is often used is the Bayes factor, which is the ratio of the marginal likelihood under the two competing hypotheses. But when we started analyzing data using this method, we found some problems, some issues here. Bayes factors were very, very sensitive to the priors we put on the parameters. And you could sort of almost get, felt like you'd almost get arbitrary results sometimes. And then the other problem is that you almost exclusively get results which support a match. And it's very well known that a simpler model may sometimes be favored to one that's more complex but correct. So that made us a little bit uneasy. And my personal opinion is that testing sharp hypothesis against a composite one in a Bayesian setting is a little bit maybe artificial. You put probability mass on one point. I don't know. It's a bit weird. So we thought, could we rethink the problem? Could we do it another way? And I know, well, a lot of people think that this is the only way to go forwards, and that's probably true. But we thought we'd try something else, and you know, to get a useful tool for the forensic scientists or the chemists to basically get a quantity to or a quantitative assessment to what they can actually see by visual inspection, but they can't express that number because in court they're often asked to give a number, a description of their certainty. And so we thought, well, let's go back to square one and think of it as follows. So the question of interest is really, are the distributions underlying these two samples, the spill and source, are they equal? Well, I mean, fundamentally the problem is, of course, that the distributions are unknown, so we can't know. If they were known, we could, of course, directly say no, they're equal, or they're not equal, or they're very close. Not equal, but very close, something like that. So what do we do? Well, we draw inference on these unknown distributions, and that results in two posterior predictive distributions. So now we have sort of two statements of belief about what the data generating mechanisms are behind these two, underlying these two samples. And so what we do then is simply we have these two inferences, and we simply compare these inferences to see whether our beliefs agree. So schematically, yes. So it could look something like this. We simply chose to take the overlapping probability mass of the two distributions, and that gives us a number between 0 and 1. And, well, partly we chose this because it seemed sort of simple and intuitive, and it treats the distributions symmetrically instead of using some directed divergence measure or something like that. And yes, well, so we did some simulations to see how this works. So we simulated 10,000 oil cases, and then we sort of analyzed them. And we had 3 different scenarios. We had identical samples, and then samples that were generated with equal parameters. So basically, they were generated from the same distribution. And then we generated samples with different parameters from different distributions. And the upper panel here is the log base factor that we got out of that analysis. And you see that they sort of pretty much blend into each other. And so it's maybe a little bit hard for the analysts or the chemists to, you know, to know what to think of this. But the agreement, predictive agreement approach that we tried would sort of quite nicely separate all these scenarios. So that felt pretty nice. And then we, of course, ran a number of real cases, and they'd been previously analyzed by visual inspection and otherwise other means too by the forensic chemists. So we had a pretty good idea of what the sort of real answer, the truth would be, but would just like to get a number for it. And if we use the figure I had on the last slide as a reference, and look at the predictive agreement here. So a positive match gives a number of 0.94, so it's pretty much up there, and it's in line with the conclusion. And probable match, it's at the lower end of when we simulated from equal distributions. So it's pretty much in line with that too. And inconclusive is not different. It's not same, but it's somewhere in between. And we analyzed a number of cases, and they were actually pretty well in line with what the previous analysis had said. And the base factor is also, it's well consistent with the results, but it's quite hard to say, you know, interpret the numbers. What's a log base factor of 7? Well, it could be a very good match, or it might not be. And as you can see, the variability is quite huge, also in the simulated results. So this seemed to give a nice number for the forensic chemists, sort of quantify what they can see by visual inspection. So that's it. I've suppressed all details actually, but the reference is there, and my co-authors, I need to acknowledge them as well, so you can find them there. But thank you very much.